Greetings, my name is Cosmo, and I've created this video tutorial to demonstrate a new bioinformatics resource that I've created called the AMASS Database. You can visit it at amass-db.org, and what AMASS is a website that provides visualization and analysis of protein structures from the protein databank. And I unify these protein structures with sequence-level annotation databases such as the UniProt Knowledge Base, the cancer mutation database known as Cosmic, or the Catalog of Somatic Mutations in Cancer, and the phosphocyte database of post-translational modifications to proteins determined from mass spectrometry datasets. So let me show you what this thing can do. If you have a favorite protein, you can search for it in this box right here, and I'm going to search for human KRAS, which is a famous protein in humans that is mutated in cancers. And this is what a typical page looks like on my database. I provide the full sequence of the protein that you've queried, and I also provide, from the Cosmic Cancer Mutation Database, mutation counts of how frequently each amino acid is mutated in tumor samples. So for example, this amino acid is present in almost 20,000 tumor samples. And the phosphocyte database tells you which amino acids are commonly modified, either phosphorylated or acetylated or ubiquitinylated in samples. And those counts indicate how frequently those modifications are seen in mass spectrometry analysis. In this table down here, I pull in information from the UniProt dataset, or the UniProt knowledge base, which indicates which amino acids are known variants, which ones have functional characteristics, such as binding sites for different molecules, or sites that, when mutated, cause different changes in enzymatic activity. And you'll notice that all of these elements are linked together. When you scroll over the sequence, you get this cursor, and if you scroll over the plots, the table automatically scrolls, and vice versa. But what my site really does that's particularly interesting is it can pull in information from the protein databank, namely any protein databank structures that have sequences that are at least 50% similar to the protein that you've searched can be loaded in. And by selecting these radio buttons, the structure can be loaded and visualized in a JMOL Java applet to visualize the PDB. And I also provide a multiple sequence alignment indicating the alignment between the protein that you searched for, human KRAS, and any polypeptide chains within the PDB that match that sequence. I also provide information on the structure, including the sequences that are in the structure, as well as ligands that are in the structure. So for example, since human KRAS is a GTPase, there is a GTP analog molecule present, and that's the name. If you click this green link here, you'll notice that it zooms in and shows you precisely where in the crystal model that molecule is located. You also get more sequence annotations as before, such as the count data and this table of annotations. However, now this information is completely interactive. By clicking on these mutation peaks, you can see precisely where the amino acids are located. So for example, this glycine residue in the native sequence is mutated to an aspartate in cancer and in the protein databank, precisely because the authors were determining the structural characteristics of this cancer mutation. I also notice that there is a phosphorylation site here in the phosphocyte database. It is a phosphotyrosine residue that is located right there. And what my website allows you to do is find out these clustered sequence features. So for example, it makes sense that since GTP binds at this site, that there's also a cancer mutation site and a potential phosphorylation site at the same location. And since all three of these elements are very close to one another, it suggests a common regulatory role. And using my tool, you can find these kinds of relationships. But what I really designed AMASS for, and the reason it's been given the name AMASS, is because my lab at the Mayo Clinic uses mass spectrometry to study human diseases. Specifically, we analyze human tissues such as muscle and liver, and diseases such as type 2 diabetes and obesity and metabolic disorders. And using mass spectrometry, as you may know, we can literally break apart proteins down to their single amino acid levels by literally blowing them apart in mass spectrometers. And by measuring the distances between the peaks, we can determine the underlying sequence, as well as modifications that occur to single sites that occur in the cell. And by piecing together these peptide sequences, we can figure out what proteins are present in the sample, and overall we can map out thousands of proteins in a given sample. And this is what typical software looks like in this particular field. The scaffold viewer, for example, that we use in our lab, gives you this kind of display and allows you very easily to figure out where the modifications are within a given protein. But the question becomes, what do these modifications do? Do any of them have a dedicated function? Do they regulate an enzyme's activity or a signaling molecule's capabilities? Now, with my website, I have an analysis engine right here. If you type in your sample name and give yourself, or type in your email address and indicate the species that your sample came from, you can upload a mass spectrometry dataset in the form of peptide sequences, where everything is capitalized. However, lowercase amino acids indicate modifications. So for example, that right there would be a phosphothreonine. And you upload the dataset, and about 30 minutes later you get analysis results that look like this. This indicates all the proteins that my system found in your sample, just like what conventional mass spec software does. And also, like conventional mass spec software, I show you the coverage of the protein along where the modification sites are located. But I also bring in the sequence annotations again, which allows you to very easily figure out the context of these modifications. So for example, in human pyruvate dehydrogenase, these two phosphoserines were detected in the mass spectrometer, and we can see from this plot down here that they are located in a cluster of phosphorylation sites that are very close to each other in the protein sequence. And if you look at the UniProt annotations table down here, several of these phosphosites are actually phosphorylated by pyruvate dehydrogenase kinase, which is the kinase that deactivates this metabolic enzyme. Now let's take a look at the structures. As you can see, there are 7 structures of this protein in the protein databank, and in one of them, my database found that serine 293 is located near a ligand called TPP. So let's take a look at that. This is the human pyruvate dehydrogenase. It's a very important metabolic enzyme that links glycolysis to the TCA cycle, so understanding modifications to this enzyme is very important for understanding human metabolism and metabolic disorders. The complex is formed of many different subunit chains, as you can see here. And there are many ligands bound to the structure. So let's scroll down and find out about these ligands. The TPP ligand that I mentioned before is thiamine diphosphate, which is a derivative of vitamin B1. And if you zoom into chain C in particular, that's the location. Now there's an extra table down here indicating the modifications that were queried from the mass spectrometry dataset, and 2 of them were tied to this particular protein, serine 293 and serine 295, which are located in this cluster of phosphorylation sites. And you'll notice that on chain C, serine 293 had a hit, namely that it was located near thiamine diphosphate. And if you click on it, you'll notice that it's actually phosphorylated in the crystal structure, because it is a common phosphosite, and it is actually well known to regulate kinase activity. And by clicking on these other phosphosites in the phosphosite dataset, you'll notice that several of these amino acids are all co-localized on this same loop of the protein. This pink loop right here. Now if you actually scroll back up to the PDB table and click on the protein databank identifier and read the abstract of this particular crystal structure, the authors actually found that phosphorylation of that critical amino acid, serine 293, in the full-length sequence, caused a dearrangement of this protein loop, which deactivated the enzymatic activity of this entire complex. So this is a perfect example about how using this web resource to analyze your mass spectrometry datasets, you can obtain an in-depth molecular insight as to how individual modifications can affect the function and structure of proteins. So, come to the AMASS website. If you've got favorite proteins to look at, go ahead and use our search feature, and if you have mass spec datasets, feel free to submit them. If you have any questions or need any help, you can contact me at this email address. Thank you, and I hope you enjoy the thing that I've created here.